Hi and welcome to our revision course for 5th years for biology. This week we're going to look at diffusion and osmosis. This was the chapter that had the most votes. So what we'll do is we'll take a look through the key areas, and what I'll do then is I'll give you some questions that you can attempt at home. I'll put up all the notes. It's a bit of a longer chapter, so we won't do all of the chapter, but what I'll do is I'll talk through some of the key areas that you need to know. So to begin, diffusion and osmosis, you need to have a definition for both. For diffusion, this is when molecules move from an area of high concentration to an area of low concentration. We get diffusion happening a lot in the world. Here's some examples. The smell of perfume is when molecules diffuse from one area to another. The unpleasant smell of a stink bomb would be diffusion. Food coloring in water. For anyone that has something like Ribena, the actual Ribena itself spreading out across the water would be diffusion. I suppose in terms of the human body, we have diffusion a lot. If we were to breathe in oxygen, that oxygen diffuses from the lungs into the blood, and it's the same for carbon dioxide, diffuses from the blood into the lungs. Okay, so again, we'll just kind of pick and choose the parts that we need to know. When we look at osmosis, it's quite similar to diffusion, but it's the movement of water molecules from a region of high water concentration to a region of low water concentration through a semi-permeable membrane. So that's the definition for it, but to try and put it into simple words, it kind of means that water is going to move from an area where there is a lot of it to where there's not much of it. And it is osmosis when it travels through a kind of I suppose a membrane there, like a space for something to travel through. Be careful when they say when water travels from a high to low concentration. That on its own is not osmosis. It has to travel through a semi-permeable membrane. Okay. Now, when we look at things like osmosis, we can actually use them to our own benefit. Before things like refrigeration existed, we used to use osmosis to preserve food. So, just mentioned there, it says bacteria and fungi cause food to decay, and osmosis prevents microorganisms from decaying the food. Two examples there are sugar in jam and salt in meat. Now the idea is that if bacteria was to land on something like jam, there is more water in the bacteria than there is in the jam, so water will move from a high to low concentration across the semi-permeable membrane. What that basically means is that water is going to leave the bacteria and go into the jam, and as a result the bacteria will shrivel and die. Now it's the same for salt. Salt and sugar have the same effect on things. If bacteria was to land on salt and meat, again it's going to lose water and it's going to dehydrate. So you're required to know one example of osmosis and food preservation. And I guess the idea here is that when we look at things like jam, if someone uses jam, they don't generally have to get a new pot of it each week. Typically what happens is that they basically can last for a long time. Okay. Now it's possible they could ask you about certain cells and what happens if you were to put a cell in this scenario and what's going to happen. Some of the previous questions in the exam papers gave you kind of an unusual scenario. There was one, I'm not sure if you've tried exam questions on this, but there's one where they used things like potatoes and talked about putting potatoes in different solutions. What you have to realize is the water will always go from where there's more water to where there's less water. Now the thing is, for things like animal cells, like our cells, they are much more at risk from things like this. So it says there, animal cells in a low water concentration. It says an animal cell in a concentrated solution will lose water due to osmosis. So basically, if you were to put our cells into a very salty solution or a very sugary solution, they'll basically shrivel up because they'll lose water. And likewise, if we put them into a very watery solution, they'll actually gain water and burst. It's also the reason why we can't take water directly into our bloodstream. If someone was to inject water into the bloodstream, the concentration difference between your blood and the water would be so great that your blood cells would actually burst. So the thing is though, a plant cell is totally different to us. And one of the main reasons is that plant cells have a cell wall, and that provides a big difference. Now, the main issue here is that the cell wall will actually provide a level of support when the plant gains water. And that is called turgor pressure. I probably have a sentence for this, yes. Turgor pressure occurs when there's pressure in the cytoplasm and vacuole against the cell wall of the plant. So you can see it here in this diagram here. The arrows represent water moving into the cell. What happens is it kind of swells up like a balloon. Now, actually, although this seems like a bad thing, this is actually a positive for the plant. The plant actually wants to gain extra water, and it basically keeps the plant upright. So if there's more water on the outside than the inside, water will move in. If you look at the cell over here, it's actually the opposite. So if there's more water on the inside than the outside, a plant cell will actually lose water by osmosis and it will shrivel up. I should have a term for that. Yeah, this term here, plasmolysis. So when a plant loses its turgor pressure, this is known as plasmolysis. If you've ever seen a plant fall over or become limp, basically, that is plasmolysis because it's lost water to its surroundings. Now you can fix that. If you looked at, I think there was an example of a question where they talked about a salty salad dressing on lettuce. The leaves will go limp, and what happens then is if you place it in water, you can reduce the plasmolysis that has happened, and that's called deplasmolysis. So deplasmolysis is a positive for the plant. Okay. The final thing here that we have to look at is the experiment in relation to osmosis. Now, what we use here is this plastic material, and that is a semi-permeable membrane. What that means is it lets some things in and not others. I often describe it to my class like a security guard or a bouncer, where they will allow some things in, but they won't allow other things in. So the idea here is that with this material, although it looks solid, it does have tiny little gaps. And what happens is that it can allow water in and out by osmosis. So take a look at this beaker here, beaker A. What's happened here is that in the whisking tubing, we have pure water. And on the outside of the whisking tubing, we have water as well. So there's water inside and water outside. What you need to notice here is that there's no difference in concentration on either side. So water can move in and water can move out, and there's no extra coming in or out. So the actual mass of this won't change. It says they're unchanged. Now if you take a look at this example here, in this whisking tubing there's sugar in the whisking tubing and there's pure water on the outside. What you always have to consider in any question is where is there more water and where is there less water. And what I want you to see here is that there's actually more water on the outside, so what happens is that water will move into the whisking tubing by osmosis. And what you'll notice actually happening is that the whisking tubing will actually gain in mass, so it will actually increase in mass. So you always have to think that they might do this the opposite way as well. They could show you that there's pure water in the whisking tubing and that there's a sugar solution on the outside. In that instance then, water would move out by osmosis. You always have to think, where is there more water? So the one on the left here is the control, and the one on the right is your experiment. Okay, so yeah, in essence the result for that will be that it gained water due to osmosis. This comes up a little bit in the Leaving Cert. It doesn't come up too often, but it does. You have to have an idea about diffusion, osmosis, in terms of things like permeability. Selectively permeable is the same as semi-permeable. That's like we were saying that it allows some materials in and doesn't allow others in. It's good to be familiar with things like the term permeable or impermeable as well. Permeable will allow all materials in and out, and impermeable will allow nothing in and out. You might be surprised to know that a cell wall is actually permeable. It allows all things in and out. You think that because it's a wall it will be quite rigid, but actually it doesn't do that. So that's the idea for this chapter. Things that are very important are the definition diffusion, the definition for osmosis, the understanding of osmosis, because really in the Leaving Cert they'll probably ask you a question that's a little bit different to what you might have seen in the book. You definitely have to know the terms turgor pressure, plasmolysis and deplasmolysis, and just be familiar with the experiment. Aside from that, hopefully you find that helpful. I'll post up some questions for you to try if you like, and if you have any questions please send me a message. Thanks and all the best.